Here we go. Hi, welcome if you are joining me for my biology GCSE revision session tonight. So I'm Christine and I have been teaching biology for a number of years, and I find that if you have a strong foundational knowledge of the GCSE content when it comes to the A level, then it becomes much easier. So I am now going to extend my offering to help those of you doing GCSE biology. So I thought we'd start tonight looking at the movement of substances across the membrane. Now, if you are studying AQA, then this is for you, but if you're also studying any of the other exam boards, then it will also be helpful what I will be teaching you. So the first thing we're going to talk about tonight is about the membrane. Now, you need to know that the cell has got a membrane and that the membrane acts as a barrier. And that barrier is between the environment on the outside, which I've coloured here blue, and the environment on the inside, which I've coloured here yellow. So we know that cells have got a cell plasma membrane that goes all the way around. We know that there is cytoplasm inside, and we know that cells are surrounded by fluid. So what we have to understand is how substances move across. And please feel free, if you have any questions, to write them in the chat and I will answer them as we go. Now, the key thing for you to note is the way small molecules can move across the membrane is what's known as simple diffusion. So again, what I'll be looking at is the key terms that you need for your exam. So simple diffusion is this spreading out of particles. Now, spreading out of particles from an area of high concentration to an area of a lower concentration. What we say when we talk about movement of particles is we say they're going to go down a concentration gradient. So if you imagine that you have a whole load of particles on one side and a few particles on another side, then what we're talking about is that we're going to have a gradient between one side and the other. So we are talking about the movement of these particles from this high concentration to a lower concentration, therefore it's going down the concentration gradient. Now, one of the things that a lot of students forget about is that particles actually have energy. They have what's known as kinetic energy. So because they have kinetic energy, what that means is the particles are able to move around. And because they're able to move around, they can move from where there's lots of them, a higher concentration, to where there's less of them, a lower concentration. So we have these particles moving, and we have to think about the factors that are going to affect the rate at which they'll move. So here I have my membrane separating my two different environments right now, and we can see that I've got some oxygen molecules and I have some carbon dioxide molecules. So there is a difference on one side to the other. So when we talk about a concentration gradient, we're talking about this movement of the substances from an area of higher concentration. So if they give you in the exam a picture, they actually expect you to count the number of molecules in the different areas. So if you were to count, we can quite clearly see that there is 1, 2, 3 carbon dioxide molecules on one side. And if we look at the other side, we have 2, 4, 6, 8, 9. So the higher concentration would be inside the cell, the lower concentration would be outside. So we know that our molecules are going to move from an area of higher concentration to an area of lower concentration. So what factors affect them is that difference in the concentration gradient. So when we talk about the low concentration, we need to think of the specific molecules. So carbon dioxide was at a low concentration and it is at a high concentration inside. So if we're looking at carbon dioxide, we have got the particles are moving from a high concentration to a low concentration. Therefore, the carbon dioxide would actually diffuse out of the cell. So it's important that you are looking at what are they asking you in a question. Now, most students jump to oxygen and the fact that oxygen was at a high concentration on the outside and a lower concentration on the inside. So in that case, the oxygen was diffusing into the cell. So that's one way in which they may ask you about the factors that are going to affect the rate of diffusion. What's that concentration gradient difference? The second thing they could do is ask you about the temperature. So I spoke earlier about kinetic energy and the particles are moving around. So you should know that if there is a increase in temperature, we're going to increase the kinetic energy of the particles. And if they have an increase in the energy, they will therefore move and they will move at a faster rate. So if we increase the temperature, we're increasing the kinetic energy, we're increasing the diffusion. If we go the opposite way, if we decrease the temperature, we decrease the kinetic energy of the molecules, therefore the diffusion rate will be lower. So we know that gases are going to be exchanged. We know that carbon dioxide and oxygen are going to be exchanged, and we know that they can move and spread out. Now they can move across, because they're small molecules, straight across the membrane. But what if we look at a molecule that can't get across the membrane? What if we look at a molecule such as water? So water is different because water cannot move straight through the membrane. So we know that we have got our cell membrane, which is this pink region in here. We know that we have got our cytoplasm on the outside, which I have represented with yellow. We have our extracellular fluid. So extracellular fluid is my blue region. Therefore, I have got a barrier between the inside of the cell and the outside of the cell. So when we look at water, because water can't get through the membrane, we have to talk about it going through what's known as a partially permeable membrane. So if we look at the number of water molecules compared to, for example, salt, in this case, I've used sodium ions as my example, we have got, let's say, 76% water on the outside and 24% salt on the outside in this extracellular fluid. On the inside, if I've got 41% salt and 59% water, there is a gradient. Now, in your exam, your teachers may have taught you about water concentration or water potential. We don't tend to use water concentration. However, we do talk about solute concentration. So if we have water moving and it's going to go down the potential gradient, we need to talk about water as diffusing from a dilute solution. Now, a dilute solution, when we say this, we're talking about lots of water. If we have a dilute solution to a concentrated solution where we have less water. So if we're going to have what's known as osmosis occur, we have this diffusion of water from a dilute solution. Now the solution is a solution because we have got a solvent and a solute held together. So the solvent is the water, the solute is the dissolved substances in the water. So if we have osmosis occurring, it is going to diffuse, the water will diffuse from a dilute solution to a concentrated solution through a partially permeable membrane. So it's really important we have these definitions. And what will happen is it will get to a point where it will reach what's known as an equilibrium. It will get to the point where there is the same number of water on each side to ensure that equilibrium has been reached. So what does that actually mean? Well, it's important that we note when we are looking at different solutions, we think about the types of cells that we have. So here I have 3 different red blood cell diagrams. And those red blood cells have been placed into different solutions. Those solutions have got a different concentration of solutes inside it. So if I have what's known as a hypotonic solution, that means that there is a higher water potential outside the cell. Water will therefore enter the cell by osmosis and it will go from a high water potential to a lower water potential. So we can talk about the high water potential on the outside and the low water potential on the inside. And if we then switch that around and talk about it with our solute concentration, we are saying that there is a dilute solution on the outside and a concentrated solution on the inside. Therefore, water will enter by osmosis. So water will get through the partially permeable membrane. It will therefore cause the cell to swell up. It's taking in lots of water, it's getting bigger, 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 bigger. And if it keeps taking in water, what will happen is that will end up with it rupturing, because animal cells do not have a cell wall. Because animal cells only have the cell plasma membrane surrounding the cytoplasm, it means that if it takes in too much water, it will get to a point where it will completely burst. So that's important for us to note. If the water concentration, so we don't say concentration in biology, we say water potential. If the water potential, and you will be given the mark if you talk about the water potential in your answer, if the water potential is greater on the outside than inside, water will enter the cell. Well, what happens if, oh, thank you for the like. What happens if you have a cell which goes into what's known as an isotonic solution? So here I have my isotonic solution. Now, it's important to note that water doesn't just move in one direction. Water is constantly moving. And so it's moving from an area of higher water potential to an area of lower water potential until we reach this equilibrium, as I said. But what we actually are talking about is the net movement. So it's important whenever you answer these questions that you do state things like it's the net movement of water from an area of a higher water potential to an area of lower water potential. And so in this case, if it's in an isotonic solution, for every water molecule that's moving in, the same number of water molecules are moving out. That is isotonic. We are at an equilibrium, and therefore the cell will stay as it should do. Now, for those of you who are already looking at homeostasis, this is really important for us to understand. If we are not maintaining our water concentration in our bloodstream, what will then result in is our cells getting too big and bursting. Or if we look at it on the other side of things, if we have a solution where there is less water on the outside than what's in the inside of the cell, the water will leave by osmosis. And if the water is leaving by osmosis, that's going to cause the cytoplasm to shrink, and therefore you see the cell pulling in and getting smaller and not being able to function as it should do. So that becomes a problem for, especially for our red blood cells, if that occurs. So osmosis is the net movement of water from an area of a dilute solution to an area of a concentrated solution across a partially permeable membrane. So what happens when we look at plant cells then? Well, animal cells don't have the cell wall. And because they don't have the cell wall, that therefore means that they have got an ability to take in the water and become quite rigid. And we call that its turgidity. So when we have our plant cells with their cell wall, which you should know is made of cellulose, this is bringing in our polysaccharides, so our carbohydrates, which are polymers. We have got this cellulose, the cellulose that's going to keep the cell shape and the structural support of the cell. So if we have this isotonic solution surrounding it, we've got no net movement. It's an equilibrium. We've got the same number going in as going out. Therefore, it stays exactly as it is. Now, plants actually can take in water because of that cell wall that allows for it to take in water by osmosis and to actually have, oh, thank you, to actually have it where you are pushing against the cell wall to the point where it can go no further. So it's a bit like imagining you having a balloon that you are filling with water, and it will be able to push out, push out, push out to a set point, but there's something stopping it getting too big. So if you've ever seen any of these videos or practicals that say to you, oh yeah, take a balloon, and if the balloon has a hole in it, put it inside another balloon and it will actually stop it from bursting. That works really well in the fact that the second layer of the balloon acts like the wall. So the wall is like the second layer, which is really strong and therefore helps the cell to keep its rigidity and allows it to grow slightly. So you can see that I have a regular cell shape here for my plant cell, but this one has got slightly bigger. So if I were to move it across, I move this across here right now, put it with the original one, you can quite clearly see that it is actually bigger. It has grown, and that, when you look at plant response, is what we talk about with cell elongation. So the cell is able to take in more water, can get a little bit bigger, but it will not burst because there is a cell wall there, and that cell wall is made up of cellulose. If we go on the opposite side of things, if we have what's called a hypertonic solution, so this time we've got less solutes inside the cell compared to outside, what's going to happen is water will leave by osmosis. So water will leave, and what you can see in this picture, and this is one that always throws students whenever they are given it in the exam, because the cytoplasm is being reduced because the water is leaving, that causes the membrane to pull closer together, because remember, the membrane is what surrounds the cytoplasm. If you've got less cytoplasm, that membrane is going to move with the loss of the water and become smaller and shrink, and so it pulls away from the cell wall. So you can see areas where the extracellular fluid have now filled in because the cell wall is permeable. The cell wall lets things through. It's the plasma membrane that doesn't, because plasma membranes are partially permeable, so water can only move through the channels that are there. So what would this look like if you were given it with regards to your required practical? So the required practical that you have to do is about investigating the effects of a range of concentrations. This is the first thing to note. We have a range of concentrations of either a salt or a sugar solution. So if you are going to look at any questions about the required practical, you need to look at what have they shown you in your table. So I've given a table of results here. I've got my concentration of my salt solution here. Therefore, because it is in the left side of my table, that is my independent variable. So my concentration of salt solution is what I have changed, and therefore is my independent variable. And I find my independent variable, if they've given me a table of results, on the left column of my table. So that is one of the hints when you're looking at exam questions and you're practicing to identify where is the independent variable. So it'll either be in the description in the exam question, or it could be in the table of results, or if they're going to give it to you in graph form, which I will show you in a minute, then that would be what is on your x-axis. So your independent variable is the variable that you are changing. So the next thing we need to look at, well, what is our dependent variable? So in this case, the dependent variable is going to be on the mass of the plant tissue. So they may ask you about beetroot, they may ask you about potatoes, they may ask you about apples. It doesn't matter what they ask you about. They're talking about plant tissue. Well, why are they choosing plant tissue? They're choosing plant tissue because the plant tissue, as we said before, has the cell wall. Therefore, it can increase its mass up to a point without the cells bursting. So it allows us to do this investigation, but we have to use plant tissue because the plant tissue is the cell wall. So what is my dependent variable? Well, this is an interesting one because my dependent variable was the mass of the plant tissue. So I've got my starting mass here, I've got my final mass, I've got my change in mass in grams, and yet they've calculated the percentage change. So why have we calculated the percentage change? Well, this links to our control variables. Ideally, with every investigation that you do, you would want to have an independent variable that you are changing. In this case, I've changed my concentration of salt solution, and I have given 5 values that I've used. So that is my concentration of salt solution. I've given 5 values from independent variable. I only want to see how that affects the rest of the investigation. So I need to control everything else. However, I didn't manage to do that. My starting mass was 1.91 in this concentration of salt solution, which was 0 millimoles per decimeter cubed. Now, I did control the surface area of my chip because I made it 1 centimeter by 1 centimeter by 2 centimeters. Now, they'll either ask it to you with regards to it being, as you can see here, this chip, or they may say that you have a cube. So it's important that you know how to work out your surface area and how that would affect the rate at which the molecules will move. In this case, because we're talking about osmosis, we're talking about water moving, we're talking about the surface area where there are going to be these channels, because it's partially permeable, which allows the water to move in and out. So because I couldn't control my mass at the start, I have different masses at the start. I need to account for that. And I account for that by working out my percentage change in mass. So my dependent variable is, if I'm given this question, the percentage change in mass. Now, one thing I would say to you is never do what I'm doing right now. The reason you don't do what I'm doing right now is because if you abbreviate in the exam, you will lose marks. I get frustrated whenever I marked these, the number of students who abbreviate for the actual exam. Don't ever do that. Even if your teacher does it, even if I do it, do not abbreviate. If you abbreviate, you will lose marks, okay? You need to write the full answer out. So whatever is written in the table, you write that in the answer line for the dependent variable. So the dependent variable, the percentage change in mass, is always, or should be, found, dependent variable, in the right side column of your table. So the right column in the table should be my dependent variable. If I'm looking at a graph, it would be on my y-axis. So whatever they give you, whether it's a table of results or whether it is a graph, make sure you're looking at the information that's been told to you so that you can give the right information and get those marks. Now, the other thing I would say is when you're talking about variables, because we love in GCSE to ask you to plan an investigation, or we love to ask you to tell us what would be control variables. So if I have controlled my surface area, what else do I need to control? Well, where did it come from? Did the potato chips that I made in my investigation, did they come from the same potato? That would be one of the ways in which I'm controlling the variable. What was the volume of the salt solution? So what volume did I actually put in when I did this investigation? So when you're doing your practice, when you're doing your investigations, you should know a set list of what variables you should go to. Have I controlled my temperature? I said earlier that temperature affects the rate of diffusion. Well, osmosis is the diffusion of water. So if osmosis is the diffusion of water, I need to control the temperature of the investigation, because if I don't, it's going to affect the rate at which osmosis will occur. How would I control the temperature? I would use a water bath. So again, make sure you know some of the bits of information that you could use for your answers. So volume, concentration, mass, temperature. There's one other that I would need to consider potentially if I am going to plan an investigation. And the other variable that I would always say to use to think about is what's known as your pH. So pH can have an effect on the structure of the membrane. So I'm going to remind myself in my head that this is GCSE level. If you're doing A level, it will go into way more detail than that. But because we're doing GCSE, understanding that pH will have an effect on the membrane structure, therefore affecting the rate at which osmosis can occur, would be something they would expect you to know and expect you to be able to say, well, what will I do to control that? I would use a pH buffer. So it's having these keywords to give for the answers to the question, whatever that question may be. Okay, so here's my graph showing those results. So I've got a graph showing the effects of the salt concentration on the mass of a potato. We can see percentage change. We can see the concentration of the salt solution. So in this case, on my x-axis and on my y-axis, I have got those results and they have been shown. So what could they do in the exam question? Well, what they could do is they could ask you to describe the data. So if they're asking you to describe the data, you would say, as the concentration of salt solution increases, the percentage change in mass of potato also changes. Is that good enough? Have I given enough detail? Well, no, I haven't. I need to be more specific, and this is where so many students miss those higher grades because they're not specific enough. So if I look at this graph, I need to be aware of, well, where is my 0 point? So here, along this point here is where 0 is. So I can see that anything above that line is an increase in mass. Anything that is below that line is a decrease in mass. So I need to be specific in my answer if I'm asked to describe the data. So describing the data would be where you're saying, hi, welcome, where you're saying that between 0 and which point here, up to there, between 0 and 0.3, the percentage change of mass of my potato has increased. From 0.3 up to 1, the percentage change in mass has decreased. So that would be describe the data, and try to be specific when you're describing that data, that you're giving examples. You must use data to back it up. And always with GCSE and even A level, 2 sets of data. So between 0 and 0.3, the potato percentage change has increased. Between 0.3 to 1, the percentage change has decreased. So that is a describe. So make sure you are looking at, in your exam, when you're doing the questions, what's that command word? Is it describe, or is the command word explain? So if the command word is describe the data, you say what you see in the graph and you give specifics. From this point to that point, I can see this. From this point to that point, I can see that. Explaining it means, well, why is it happening? why is it that these ones have increased in mass, and why is it that these ones have decreased in mass? So then we have to think about, well, where would the water be going? Would the water be entering or would the water be leaving the cell? So if my mass has increased, I can therefore explain that in that water, let me change colors, 1 second, try and keep this consistent. My water has entered the cell. So water has entered the cell. If I go to decrease, I can say, well, it's decreased because water has left the cell. Great, I have maximized my grade. I am getting 100% in this question. Unfortunately, that's not the case. And again, this is where a lot of students miss out those extra marks because they don't go into enough detail. So, water entered the cell from 0 to 0.3. Why? Because there was a higher water potential outside the cell. So I know that the outside of the cell, there was a higher water potential outside compared to inside. And I know that if I'm looking at the bottom region, why did it lose mass? That's because there was a higher water potential inside the cell. Therefore, the water left the cell. So if I'm looking at my cell, water is leaving my cell when we're in the bottom region. If I'm in the top region, water is entering the cell. So what can you tell me about this point right here on my graph? Well, there is no increase in mass and no decrease in mass. Therefore, that must be the isotonic point. So that must be the concentration inside my cell. So when you are looking at a question that is asking you to either describe the data, say what you see, if it's asking you to explain the data, you have to give the science that is going on. Why did that happen? So the key thing about explain questions is, why is it happening? Yep, what would you like to ask? Please do ask away. Sappy, what's your question? Sorry, I'll just keep talking and explain things. But if you do have questions, the whole point in these sessions that I want to do is to get you to actually ask questions so I can explain and help you understand. So please do ask away. I'm going to carry on, but if your question comes up, I will come back to where we are at, okay? So, what else could they ask you in the question? Well, they could ask you to describe how the students could find out if the data is both repeatable and reproducible. So this is where they're bringing in some key scientific vocabulary. Do you understand what it means if something is repeatable? Do you understand what it means if something is reproducible? So could you give a definition for that? So when it comes to a question like this, what they're actually assessing you on is your understanding of key scientific terminology. So what does it mean if it is repeatable? So it gives it away with a word. If the data is both repeatable, it means that you are able to repeat the experiment. You are repeating the experiment, and you are repeating the experiment and getting the same results. So it's important that you are going to repeat the experiment and you will hopefully get the same results. But by repeating it, you can identify any anomalies. And anomalies are data that doesn't fit the trend. So if you've got data that doesn't fit the trend, then that is an anomaly. So by repeating the experiment, what we would normally do through that repetition is we would want to calculate the mean. And by calculating the mean from using those repeats, we would also be able to account for any anomalous results. Now, if you go on to study A-level biology, you're going to learn more about not just about the calculation of the mean, but also something which is known as the standard deviation. Now, the standard deviation you will have seen, but at GCSE we never talk about it in the term of standard deviation. If your teacher has, then well done. I'm really proud of them, because that is important. The more we drip feed that knowledge into you, the more it makes sense. But when you do a graph and you mark the mean with, let's say, can be a cross, can be a diamond, can be anything. But this is the mean. What we would normally do is add in error bars. So error bars allow us to see the variability in our data. So those error bars, you will learn about the fact that that is showing what's known as the standard deviation. But that's if you go on to study biology at A level. So if they ask you how they could find out if their data is both repeatable, repeat it. Repeat the experiment, keep repeating it, identify any anomalous results, ones that don't fit our pattern, and we can do that through the calculation of our mean. Now, what does reproducible mean? Well, reproducible is slightly different. If someone else, so if another student were to carry out your investigation, now to be able to carry out your investigation, they would need to have a method that they could follow. So as I said to you before, you only want to change your independent variable. You want to measure your dependent variable. You therefore need to control all other variables. And so because of that, the method that you produce should be a valid method that will allow for someone else to pick up your instructions and to do exactly as you did. So if it is reproducible, they are going to conduct the experiment. However, what they might do is they might use a slightly different procedure and see if they get similar results. So if they were to use a different method, so they could use exactly the same method as you, and therefore they are repeating it. But if they are using a different method or using different equipment, would they still get the same results? So what you're doing is you are looking at, are they able to reproduce the data that you got, showing that if you carried out this investigation, that trend would follow as you would expect. So if it were, for example, using a different beetroot, using a different potato, do they get exactly the same set of results? Is it reproducible? Now, another thing they can do in the investigation side of things, so they do expect you to know how to do that with plant cells. Sometimes they love to throw in a question showing you a model which would be, for example, showing the process of osmosis. So if we were to use visking tubing is a special type of a material which actually has holes in it. And because it's got holes in it, it's going to model that partially permeable membrane, because it will only allow some molecules out or in if they are small enough. So if they are too large, they won't allow it out. So in this case, what we have inside is we have our viscin tubing, we have a solution which I've coloured purple, and a solution which is blue. So we've got solution W and solution X. Now, the capillary tube is going to allow the liquid inside to move up or down, and we are able to then measure where its starting position is and its ending position. So in this case, this is the start, and the start was 10.5 millimeters. This was the end after it had been in the solution over a period of time, and it was 26.5 millimeters. So if that's the case, if the coloured liquid has moved up the capillary tubing, can you explain why? Can you explain the difference between solution X to solution W? Well, if the liquid inside has resulted in it moving up, as we can see quite clearly here, that would tell me that the liquid from solution X is able to move into the viscin tubing. And because of that, that is causing the liquid to rise up higher within the capillary tube. So if we understand that, we can therefore make a conclusion about what's going on. We can conclude that solution X must have a higher water potential than solution W, because the water has moved into the viscin tubing. Therefore, because the water has moved into the viscin tubing, it had to be at a higher water potential outside than inside. And that therefore tells us that our solution W had the lower water potential. So if they ask you to draw a conclusion and they've given you a set of results, what you need to be able to do is think about, well, what have they told me? It's a partially permeable membrane. So therefore it only lets some things through. Why? Well, if it's partially permeable, it's got pores, and those pores are only going to allow small molecules. So it could be that this is a starch solution, for example, and starch is too big to get through those pores. And because it's too big, it can't get through those pores. It therefore can't leave the viscin tubing, but it will have an effect inside, or will it? This is where you have to understand that whatever is inside has to be something which is large enough to not get through. So it needs to be large enough to not get through, but also needs to affect the water potential, and therefore needs to be soluble. So we need something that's soluble, so therefore we would use salt or sugar and not starch. However, they may use the viscin tubing with starch and look at iodine test and looking at the effects of amylase. So this is one way in which they could link this together with a partially permeable membrane, but instead of it being the movement of the water for osmosis, they may be looking at the movement of, for example, sugars, which leads me on to my next thing. So if we're looking at the movement of substances across a membrane, We know that diffusion, small molecules, will move down the concentration gradient. We know that osmosis, we know osmosis is where you have got the water moving down the concentration gradient through a partially permeable membrane. So what is active transport? Well, active transport is where we have the movement of substances, and that movement of substances is going to be against the concentration gradient. So if we're thinking about, as we showed before, our molecules and we have a concentration gradient, we know we've got lots on one side and less on another. So we have a low concentration on one side, a high concentration on the other side. If we're going against the concentration gradient, we're going up the hill. It's much harder. And because it's much harder, it means that it requires energy. If there's no energy, then there is no movement of the substances against the concentration gradient. So if it is a passive movement, we've got diffusion, we've got osmosis. If it is an active process, we require this energy to be released for the movement against the concentration gradient. Now, the energy that is required is in the form of ATP. And if you go on to study A-level biology further, you will learn about that structure and you'll learn about how it releases the energy. But for GCSE, what you need to know right now is that active transport requires the energy, and that ATP molecules come from the respiration that occurs within the cell. So why is that important? Well, really interestingly, plants, we've been looking at our plant cells a lot today. The plants have got the cell wall. The plants need to take in water, because in another lesson we'll talk about photosynthesis, and photosynthesis has water as one of its reactants. So therefore plants always need to be taking in water. So therefore they always have to have water moving into the cells by osmosis. And so therefore the root hair cells have adapted. They are specialized in their role in the uptake of water. And they do this through having these long extensions. So a root hair cell is a cell in the root which has this really long extension, which is why it's called a root hair cell. We're not talking about the hair in your head. We're talking about these cells which are found in the roots of plants in the ground, which are there to take up water. So by having this hair extension, this long extension, what it is doing is increasing the surface area, increasing the membrane at which the water can be taken into the cell. But the only way we can get water to move into the cell is if we have got one, pores to allow water to move in, but two, if we have a concentration gradient. So a root hair cell is adapted because it has lots of mitochondria, and mitochondria provide the energy for active transport. And what needs to happen is the mineral ions will need to be moved into the cell against their concentration gradient. And because they're moving into the cell against the concentration gradient, what that means is that's going to lower the water potential inside the cell. So that water will move into the cell through the pores, and the water is moving down the water potential gradient from a high water potential to a lower water potential. Now, technically, it's actually hydrogen ions that are pumped out and they move in. But for you, you need to just talk about the active transport pumping mineral ions against their concentration gradient. So we know that they're being pumped against the concentration gradient. Therefore, we know that if it's against it, it has to be from a low concentration to a high concentration. So that will therefore change the water potential, and that will therefore allow water to move in by osmosis. So energy is required because we are obviously doing active transport. And then the last thing I'm going to finish on tonight, and next week I'll actually look at respiration and link that with how this is needed. But blood glucose concentration is maintained within our bloodstream within a very narrow limit. So if you've done homeostasis, you've learned about your blood glucose concentration and the fact that we need to control that through the use of hormones. Well, glucose is absorbed by active transport. It's absorbed from our intestines into our bloodstream against a concentration gradient. And because it's going against the concentration gradient, we know that energy is required. Why is it going against the concentration gradient? Well, it's going against the concentration gradient because there's already glucose circulating in the bloodstream. The concentration of glucose in the bloodstream is greater than the concentration which is inside our intestines after we've digested our foods. So therefore we understand that it's through a process of active transport. But again, if you go on to study A-level biology, you'll learn so much more about what is actually being actively transported and how that is used to move the glucose against a concentration gradient. I hope you have enjoyed tonight's session. I hope you found it useful. Please do click on the like button if you have. Please do subscribe to my channel. I want to keep making more of these videos and keep helping you more as much as I can. And so therefore, thank you for joining me. Thank you for asking questions. And I look forward to seeing you next week if you come back again.